Hello, everyone. My name is Hyeonwoo Kim. I'm a assistant professor at KAIST in South Korea. Here I talk about exercise and space biology. Especially, I'd like to discuss about how we can look at exercise and molecular levels or biochemically, and how we can use that factor or the information in space biology. As you know, exercise has several beneficial effects that is not limited to the muscle, but also many beneficial to the cardiovascular system, brain, and metabolic organs, also bone and hormonal system. Also, that can be divided upon exercise type. There are 4 types of exercise upon NIH category. The first is endurance exercise, which includes walking, jogging, or dancing or swimming. And that actually improve the heart, lung, and circulatory system. Also, it is also beneficial in the cancer, and there's impact our brain. Resistance exercise, which is the lifting weight or push-up or etc., and that actually improves your muscle and also enhance bone mineral density. And other 2 types of balance exercise and flexibility exercise. But I'm going to mainly discuss about endurance and resistance exercise. As they are very beneficial, it can be also related to pathological condition, such as the lowered incidence of the pathology. Here, this figure shows that exercise group has way better than sedentary condition in terms of the percentage of the incidence. For example, dementia, hip fracture, and depression are lower. And also mortality and the cardiovascular disease, diabetes, and the cancer also show very similar effect. That means exercise can be the therapeutic application to many pathological conditions. How about space? As you know that space has some issue. That is the bone loss and muscle loss. Even though we are dreaming to go to Mars, because bone loss and muscle loss is probably not easy. And that can be from the microgravity radiation and many other extraordinary circumstances. However, here I'm mainly focused on microgravity, because the microgravity actually caused many issues, such as weakened mitochondria and the change in extracellular matrix and many other gene expression change, and that can lead to bone loss and muscle loss. And people try to use the exercise to improve that bone loss and the muscle loss as exercise impact the bone and muscle, but it is not easy. So what's the matter between microgravity or space biology and exercise? Let's look at the exercise. There are at least 2 major components in exercise, which is mechanical stimuli and the biochemical communication. And this combination actually impact our body, and the combination may matter in this case. For example, in the NASA, they use the mechanical stimulus to improve the bone and muscle mass. By that, mechanical stimulus, mechanical stimulus actually improve the muscle loss or restore the muscle loss or bone loss. So they're giving artificial mechanical strength to their body. But how about the biochemical communication? Because there are known factors to understand biochemical communication, but still it needs more study to understand. But we can discuss with the known factors. As how we can look at the biochemical action, because muscle mediate exercise and responsible to exercise action, we can look at the muscle first. The muscle actually do action for our performance in terms of exercise that change something in the muscle cell that we can call several molecules, and we try to understand how the molecular action impact our exercise effect. That can be also, as you mentioned, endurance exercise can be studied, or resistance exercise can be studied using the many mouse model system, and you can dissect how the molecular change happen each exercise, and you can describe how exercise impact our body. One studies came from the Prof and the other several researchers. For example, endurance exercise change certain transcription coactivator, which is called PGCon alpha. That is known to be a therapeutic factor in fat, but later the other researcher and Prof showed that actually endurance exercise increase PGCon alpha expression in skeletal muscle and seems to be involved in some gene expression change. So Bruce Spiegelman group may generate the alpha trenching mouse specifically in the muscle and look at what happened to muscle. As you can see that the white mouse, this is a muscle, and you can just focus on the color, and you can see that this trenching mouse has more red in their muscle. That means without exercise, but change or overexpression of alpha by itself can change the muscle type as type 1 muscle type, which is more intravenous exercise-induced muscle fiber type. And that means alpha somehow recapitulate intravenous exercise effect. Also, there's more, like mitochondria can be more made, and the vascularization enhanced by that alpha overexpression. More interestingly, that is not limited to the skeletal muscle, even if the expression is in the skeletal muscle, but also that alpha overexpression in muscle actually impact the pancreas to increase insulin secretion, and they could be involved in the diabetes phenotype or therapeutic approach. So that means some alpha actually change something in the muscle and then impact to pancreas through tissue crosstalk. And that means something can be secreted from the muscle upon alpha action. So later, Bruce Spiegelman group studied what could be the secretive factor. And then as expanded area, we can think about that. Exercise somehow impact the muscle, and muscle is responsible for that kind of exercise action. And that muscle secretes something, and that secretive factor may go to the other organs to impact our body. That gives understanding of the system effect of the exercise. Also, if we know that factor, we can use the therapeutic approach to many diseases, which can be relieved by exercise. So the first thing the Bruce Spiegelman group discovered is the irisin, which came from the FNDC5 or type of membrane protein, the skeletal muscle. And their gene expression actually increased upon endurance exercise, and later that type of membrane protein cleaved and released the soluble fraction, which is named as irisin. And that irisin protein get into the blood system and they go to other organs. The one thing the Bruce Spiegelman group discovered is that iris impact to the bone. This is a genetic mouse model, and this is the loss of function of the irisin, and then they show that actual iris involved in the bone resorption upon ovariotomy, which is a mimic of the menopause, which has serious osteoporosis situation in human disease. This is bone stained lumbar vertebrae and stained with the bone mineral with this staining show that the wild type after between the sham group or ovariotomized group shows a very different bone mineral density as known. Like sham group have a stained area, which is bone mineral area. You can see that there are bone mineral area. However, ovariotomized group, or which is mimic the menopause, which has osteoporosis, you can see that many bones are gone. That means that ovariotomy drives bone resorption. However, in the lack of the irisin or the precursor FNDC5, you can see that the bone mineral is both either sham group or ovariotomized group that all have that bone mineral in that black bone. And the quantified data also show that in the ovariotomized group, this irisin knockout mouse model has more bone volume and more trabecular number. That means some irisin is involved in the ovariotomy-induced bone loss. To confirm the irisin function, the transgenic irisin mouse in the skeletal muscle specifically has way less trabecular bone volume than wild-type mice. So both talking about irisin contribute to bone loss or bone resorption. That means probably that irisin could be the kind of the therapeutic target if you block irisin action, if they matter in the space or microgravity, which is not yet tested. So that will be a very interesting project to understand whether irisin level can be changed upon the space by our space area or microgravity. And then if we block that irisin, that can be a kind of therapeutic approach to the bone loss. And also, furthermore, there's more interesting outcome that iris actually contribute from research directly working on osteocyte, osteoclast cell, and then that also involved in the thermogenetic expression in the fat cell, so it can be involved in energy expenditure. Also, that all be did by integral alpha V family and the CD81, and then this is a receptor and cofactor system. So that based on the mechanism, we can approach to the understanding of irisin action in space more in detail. The second protein or exercise in this hormone is the nutrient, which is another endurance exercise in this myokine. That also expressed in the muscle, and then that also their expression can be mediated by physician alpha, and then actually working on neuromuscular junction formation also drive the slow motor neural identity, so they can be more favorable toward endurance exercise induced muscle fiber. It's a very interesting muscle-specific nutrient mice have better exercise performance than wild-type mice, as you can see here. Also, that too are protein hormone from the muscle, and they can be induced by endurance exercise. But also one of the metabolite hormone is identified, which is the beta amino acid butyric acid, or here L-BIBA, which is also induced by endurance exercise, and then also physiological factor working on the production and secretion of L-BIBA. And when L-BIBA actually working for bone and then fat, the first thing is that L-BIBA is kind of working on preventing the bone loss by relieving the reactive oxygen species stress in osteocyte. And osteocyte is a key bone cell to maintain the bone homeostasis by sensing the mechanical stress and the biochemical action, and then also giving a signal to osteoblast or osteoclast cell to maintain the bone homeostasis. Also, interestingly, L-BIBA also working on the thermogenesis in fat. So that means, as in the microgravity, there is the increased ROS level, and if we can use that L-BIBA to suppress ROS level in the muscle, it can be also useful to restore or maintain the muscle function or bone function. So that can be another therapeutic approach. The other one is that all the three which I mentioned is by induced by endurance exercise, but now this is the one which is involved in the resistance exercise that is called myostatin, which is discovered in the muscle, and actually that is the secreting factor from the muscle but suppressed by resistance exercise. And then that is a negative regulator of skeletal muscle mass. This is the figure from the paper, show that compared to the wild-type mice, this knockout of the myostatin actually have much bigger muscle. That means some myostatin involved in the muscle size and the hypertrophic action. And also that also involved in the somehow implied factor for research in the rheumatoid arthritis. Also, people now discuss about space biology with myostatin as that spaceflight seems to increase myostatin gene expression and protein level in the skeletal muscle. That means probably that may work for preventing the bone growth or muscle growth. And there's a lack of the myostatin signaling actually decreased muscle loss of the mice in space. So then that can be a very good therapeutic approach, which is maybe the also good approach in the future study. So as summary, each exercise has its own effects on our body, and biochemical understanding of exercise requires the utilization of space biology. The application of that type of exercise hormone can be therapeutic application to bone and muscle loss in space. Also, I just mentioned fuse, but also you can see many other factors, including interleukin-6 or other cytokines and the other new hormone discovered recently. What I'm trying to understand is the always perspectives as now we understand exercise at the level of the molecular level and the molecules, and we try to understand biochemically how exercise can be defined and how we can use that factor the many diseases, including the space biology. So in our lab, we try to understand how different exercise challenge impact our muscle, also maybe bone as well, in terms of stem cell, immune cell, and the regeneration system upon their heterogeneity and the metabolism. So that we can understand how bone and muscle responds to that exercise. And then we can also use the secreting mouse to understand what muscle secreting factor and the bone secreting factor or myokine or osteokine can be identified newly and then characterized. And then what's the impact on our body? And we can use that kind of information to understand how we can use that kind of factor in space biology. Here, bone and muscle loss can be mediated by space travel, and we can mimic that microgravity during the random positioning motion, and we can understand how that can happen at the cellular level and the biochemical level, and then we can target that kind of effect in vitro and in vivo system eventually, and we can understand how we can use that exercise hormone can be relieve this type of the effect. Thank you for your listening. And I just opened my lab after training under Bruce Spiegelman at the Dana-Farber Cancer Institute and Harvard Medical School. And I opened my lab to understand exercise biochemistry, and they also use that information in space biology. Especially, I'd like to thanks to Joshua Cho at University of Technology, Sydney, who is a good collaborator. We are trying to understand exercise, and we understand the metabolism in the space biology. Thank you for listening.